Dr. Gauri, you may start with the introduction.
Sure, sir. Thank you, Patil, sir. Good morning, one and all. I welcome you all on the 14th day of 21 days training program. Our today's speaker is Dr. Rajendra D. Patil, who is veterinary pathologist, and he is also my teacher from UG program. He is known for his deep knowledge in the field of veterinary pathology. Thank you very much, sir, for accepting our request. Now, I would like to take the opportunity to formally introduce him. Dr. Rajendra Patil has completed BVSc and AH from KNP College Veterinary Science, Shirwal, District Satara in year 1999. MVSc in Veterinary Pathology from College of Veterinary and Animal Science, Parbhani in the year 2001 and PhD from Deemed University IVRI Izzatnagar, Bareilly in year 2007. He worked as assistant professor in the Department of Veterinary Pathology, College of Veterinary Science, Rewa, Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur for 1.5 years. And at present, he is working as assistant professor in the Department of Veterinary Pathology at Dr. G.C. Negi College of Veterinary and Animal Sciences, CSK HPKV, Palampur, since February 2009. He has 13 years of experience in the field of teaching, research, and extension. He is a recipient of World Poultry Science Association grant from the Master Research on Microtoxicology in broilers and Institutional Senior Research Fellowship during the doctoral program on ochratoxin A-induced developmental toxicity in rats. He has published more than 70 publications in the Journal of National and International Repute, and he has H-index of 50. His research area of interest are experimental toxicopathology, animal oncology, and pathology of infectious disease of livestock, including poultry. Thank you very much, sir. I request Dr. Patil now to please deliver the lecture. Thank you, sir.
Thank you, Dr. Gauri. So, dear participants, first I want to welcome all of you, and I'm going to talk about the recent advances in the diagnosis and amelioration of mycotoxicosis in animals. And you know, the mycotoxicosis is a problem, is not for our country, but it is a worldwide problem. And the diagnostic challenge and the amelioration strategies, that is a researchable issues. And till date, although it is a old topic, but till date, a lot of research work is going on regarding this topic. So that's why I have chosen this topic, and we are going to interact about this. What are the mycotoxins, how they are causing deleterious effect or they are causing mycotoxicosis in animals. Then what are the diagnostic methods or the newer diagnostic techniques, and the amelioration aspects we are going to cover today. So regarding the mycotoxins, these are the low molecular weight, biological active, highly toxic secondary metabolites of the mainly produced by the toxigenic species like Aspergillus, Penicillium, and Fusarium. These genera, the number of species, they will produce these mycotoxins, and these mycotoxins are ubiquitous. These fungi are ubiquitous in nature, and you will find these fungi everywhere. And almost it is estimated that all the food commodities are contaminated with the mycotoxins. Not a single commodity is having exception, and all the food commodities, those are having the mycotoxin contamination. Among these aflatoxins, citrulline, trichothesis, and fumicin, these are the major groups. Otherwise, more than 300 mycotoxins, those are characterized, or those are identified in the nature. And more than that, those mycotoxins, till people are working about their action mechanism and their pathology or their deleterious effects. And that's why it's a problem. It's a worldwide distribution, and that's why it's a problem. And till day, a lot of work is going on regarding these aspect, as well as the diagnostic technologies, to develop diagnostic technologies regarding the detection of these mycotoxins in food and food products. In India, because of this, our country, the tropical climate, that is hot and humid climate, and that hot and humid climate will always favor the growth, mold growth, and that mold growth will always uh, liberate these toxins, and these toxins are biological active. Once they enter into the body of the animals or humans, they will produce deleterious effects, and that's why these mycotoxins, they are concerned because they have a wide toxicological spectrum. They include acute toxicity, chronic toxicity. Because the dose of any toxicity, when you look into the animals or in humans, the dose and the duration, these two are very important for the kind of toxicity or to see the deleterious effect on the body. So they are having acute toxicity. If the dose is more, large amount of toxin consumed through diet by the animals, then they may produce chronic toxicity. If small amount of mycotoxins consumed by the animals for over a long period, so these mycotoxins have cumulative effect, and they will bioaccumulate within the organ system or within the fat fraction, and ultimately they will produce the chronic toxicity. Then besides, they are having the immunosuppressive action, and immunosuppressive action is the most important, particularly, and that is in the poultry industry. And this immunosuppression, it is caused by the mycotoxins through diet. If small amount done by the through feed by the animals, they will cause the immunosuppression, and the immunosuppression will always for the infections, individually for the infection. Then beside that, they will also exert teratogenicity, that is body, then carcinogenicity, that is the liver cancers, then mutagenicity and genotoxicity. Those are the other effects, and that's why they are more concerned. Besides these, they are possessing the number of properties. I mean, that most important property is heat stability. And these, it is suggested, a lot of literature suggested that at higher temperature also these mycotoxins we can't destroy. And that's why they are very challenging to destroy these mycotoxins or to completely eliminate these mycotoxins once the feed of water is contaminated with these mycotoxins. Even they are having the lipophilic in nature, and they will bioaccumulate, and they will in the fat fraction they will deposit, and they will enter into the food chain in the form of residue. Whatever meat, milk, or eggs we are consuming from the animal products, and those are also as in the form of residue. Those are also affecting to the humans as well, and that's why they are more concerned about the public health significance. Again, besides having long biological half-life, and long biological half-life makes the different mycotoxins, the different pathology and the pathogenesis or mechanism of producing the symptoms or the lesions. Long biological, particularly mycotoxins like ochratoxin. It is suggested that around 30 days is the biological serum biological half-life in the humans, and that's why in endemic countries like Balkan endemic countries or Scandinavian countries, where the high prevalence of ochratoxin and as well as and by the endemic nephropathy that is well reported and well studied by the people over the decades. Besides that, these mycotoxins, they will cause huge economic losses, and the huge economic losses because of the toxicity or toxicity of chronic toxicity, and that's why the production losses in terms of like food industry, the poor FCR, food feed converter. These are the problems with the animals and poultry. Then classification of mycotoxins. Mycotoxins are basically classified on the clinical manifestations, and they will target organ where they will primarily, once they ingested, and these are the classified on the basis of the experimentation of these mycotoxins. And mycotoxins, they are categorized as a hepatotoxic when the hepatotoxic mycotoxins like aflatoxins, chloroquine, and luprotoxins, fumicins. Fumicins also primarily hepatotoxic mycotoxins. Then nephrotoxic mycotoxins, they include ochratoxinase, citrulline, osporin, quinones, then vimelin. Then neurotoxic like fumicins, patulin, etc. Immunotoxicity like the aflatoxins, ochratoxinate, trichothecin, citrulline, then photosensitivity action of petroglyphin, These all are in estrogenic, zebranon is estrogenic mycotoxin. Then teratogenicity, mycotoxins like aflatoxins, cyclamin, fumonisin, trichothiocin, these all are teratogenic and they are causing birth defects, and that's why they are more concerned for the pregnant animals. And the cytotoxic like trichothiocins, they are directly causing cytotoxic effect. So regarding the, if you see the mycotoxins, all the food and the feed and food commodities, those all are, and the among that the major is the maize. Maize is a mostly contaminated for the mycotoxins. And in Indian scenario, if you look to the Indian scenario, these mycotoxins, these all are the four mycotoxins are more prevalent in our country, and among that the aflatoxin is the most important. Aflatoxin, cyclamin, and the trichothiocin. These are important. Although nowadays and other reports of other mycotoxins, those are also available. Then the aflatoxins. Regarding aflatoxins, it is a main produced by the Aspergillus flavus and Aspergillus parasiticus, and the aflatoxin B1 is the most toxic compared to other members of this family, like B2, G1, G2, M1, and M2 are the secreted through the milk. And these aflatoxins are primarily hepatotoxic, as well as it is a potent carcinogen, and the National Agency for Research on Cancer in 1993 they categorized it as the potent carcinogen or the ultimate carcinogen, and they categorize it as a potent carcinogen, and it will produce tumors in animals and humans. The immunosuppressive action is very important, and particularly low level of aflatoxins to diet taken by animals or human beings. So it will always produce the immunosuppression, and immunosuppression is always lead to susceptibility for the number of infections of the individual. Then the teratogenic, we have also seen in our laboratory that the aflatoxins causing not only aflatoxins, even citrulline, they are teratogenic and they are producing the defects, the malformations, cross visceral and skeletal malformations. And this disease in 1960s, the more than 1 million turkeys they died because of this disease. At that time, we don't know the that's why they on the turkeys disease. Even in dogs, the liver was the predominant feature at the time of postmortem, the mortality, and that's why the takes disease. The LD50, those are suggesting the species susceptibility and the LD50 values, and the suggesting that the ducklings are the more susceptible than the particular rats or the other species. Those are resistant. Then grossly, what you will find besides the depression, growth rate, decreasing growth rate, in the animals and birds, grossly when you will perform the postmortem, you will find the liver is a yellow, enlarged, fatty, and with the and the chronic toxicity, you find the tumors, particularly those are well reported in ducks. Then the swallow and large, and the organs will be microscopically you can find the degenerative protein changes, because in most of the mycotoxins, although they are target organs, but the degenerative and necrotic changes are the typical pattern you will find in most of the mycotoxins. But the typical lesions like bile duct epithelial hyperplasia, particularly in this acute chronic toxicity and the fatty change, these are very typical, or I can say somewhat diagnostic. In most of the seen in the rabbits, as in poultry, even in the rats, these changes are similar of the changes you will find at the infiltration of the uh, uh, inflammatory cells. It is a secondary target organ for these macrotoxin aflatoxins. It also produce degenerative necrosis in the organs. So this picture, gross basis, when at the time of this is on the poultry liver, and you can see the pale yellow and enlarged fatty liver, and this is a characteristic. And
I think our resource person got disconnected. Let's wait and see if he joins back.
Please wait, everyone. Please wait.
Dr. Gori, I tried to call him up. Can you call him up and ask him to join again? Because
Yes,
he
sure, sure,
disconnected the
sure.
call.
Okay. Majid sir, he is going to rejoin again.
Okay, ma'am. Okay.
He has joined, sir. Dr. Patil, please share your slides again. He's again.
And turn on your camera as well as your
He has left the
unmute.
meeting again. I guess his net is not working. I'll
Yeah,
just
I think so.
check again. Okay. Okay, he has joined again.
Welcome back, sir.
Welcome back, sir.
Can you share your screen again? Can you also can you unmute? Your microphone should be red. Can you click on it so that it turns white again at the bottom?
Okay.
You are unmute now.
Now it's audible?
Yeah, you are audible now.
Yes.
Can you share your screen again?
Okay.
Present now.
Okay.
Choose entire screen option the same way.
Yes.
And then click on the screen and the share button should turn blue. And click on the blue share button and it will start. It is starting.
It's visible now?
No, not yet. Did you choose the entire screen option? Yeah.
It's
Now
visible.
Now it's coming?
Yeah. Perfect. Can you start the slideshow? And participants, can you help us where the sir was on the slide so that we'll move to that slide?
Fifth or sixth slide, I guess. So please move forward one by one.
Hello? Can you move ahead in the presentation? Can you turn off your video? I think your network connection is slow. I think his network connection is slow.
Okay.
No, not like.
Now it's.
Yeah. Please go ahead.
Where, sir? I was exactly. I don't know.
Sir, in the Chrome, can you end the slideshow first? End the slideshow. Right click and end the slideshow first. Right click on the slide and end the show. And go to Chrome. Turn off your camera so that your bandwidth is better utilized.
I guess his net is not working properly. That's
No,
why.
so that's why I'm asking him to turn off his camera.
So
He
I
hasn't.
will switch to another system. I will switch to
No,
another
just
system. Wait a minute.
no,
Okay,
system
okay.
is not the issue. Network is the issue.
Network issue is the problem, basically.
Sir, can you go
I have
to the
another
Chrome? Sir, follow
landline
the
connection
steps,
I'm
please.
having. This
Sir, I'll tell you what to do to better utilize this connection. Sir, can you end the slideshow?
Okay.
Right click and end the slideshow. Yeah. Turn off your camera. It should be red. At the bottom, your camera icon should be red. Turn off. Click on it. It should be red. So that your bandwidth is not utilized in video. Yeah. Can you click on the camera icon at the bottom?
Okay.
And it should turn red.
Okay. Then.
Have you turned off your camera? Yeah. Now
Yes, sir.
go to PowerPoint and start your share your screen. Again, share your screen. Present now. Share your entire screen.
Okay.
Now, since your camera is not working, it's not taking that much of bandwidth. Your audio would be fine.
Okay.
Yeah.
It's
Open
visible
the
now?
PowerPoint and start with the slide.
No, sir.
It's loading.
Not yet.
It's loading.
Just sir, once. Now it's visible?
Not yet. Have you chosen the entire screen option?
Yeah.
You stopped it, I guess.
Again, I am trying.
Okay.
Shall I switch to
Yes.
another?
Go to PowerPoint. No, it's working now. It's visible now. Can you go to PowerPoint?
Okay.
Generally speaking, right now your connection is slow. So that is the issue. The machine is not the issue. The network is the issue.
That's why
I'm having
he's
a
saying
landline
to switch
connection
over to
I'm
the
having.
landline system.
If
This
it is
was from
possible to
mine.
get a better connection, that would be great.
Sure, sir.
Yes,
You
I
go
will try.
ahead with the second system of connectivity.
I thought some frequent electricity cut. That's why I have not chosen this landline connection.
Right now it is visible, sir. You can proceed with the presentation.
It's now visible.
Yes, sir.
Yes,
Next
yes.
slide, sir.
Next, Dr. Gauri, can you tell me on which slide
Yes, sir.
from
Yes,
which
sir.
I
Please,
would...
please, next slide, sir. Next slide.
Aflatoxins we have covered?
So it's not actually connectivity. I guess this one. Yes, sir. You may start here only. Microtoxins.
Okay.
Yes, sir.
Okay, no problem. I'll start. Okay, no problem. So regarding the next important mycotoxin group is aflatoxins, and as you know, this mycotoxin is mainly by and these mycotoxins, you know, the fungi, the toxigenic fungi belonging to Aspergillus and Penicillium, they produce single species can produce multiple toxins. multiple fungi, they may produce a single type of toxins, and that is the specialty of this toxinic this family. Then the ochratoxinae is chlorinated and more toxic than the other family members like ochratoxin B and ochratoxin C. And its chemical structure is filamine linked by an amide bond to dihydroisocombined moiety. And the target organ is the kidney, and that's why it is most important nephrotoxic mycotoxin, and it is not concerned with the animals but in humans. Particularly endemic regions, as I told, in Balkan and Scandinavian countries, where the endemic nephropathy, and particularly in human population, they die because of these nephropathy. The correlation with disciplin along with the mycotoxin, these are the main reason through diet, the mycotoxins, and they are affecting this nephropathy. Even the developing nervous system, and the developing nervous system is also very important target site for the mycotoxins. And during the teratogenicity, even in my PhD program, I have worked on the teratogenicity of this ochratoxin, and I have seen it is classically affecting, not only affecting to the kidney, developing nervous system. I'll show you some pictures. Then the endemic nephropathy, as I told, besides the Balkan nephropathy in humans, it is also causing post-endemic nephropathy in the pigs. That is also another important animal which is susceptible for the ochratoxins. Besides the dogs, pigs are very important. Even chickens, those are susceptible for these mycotoxins. And a number of outbreaks are reported time to time in India, among the poultry, even the Pashmina goats, buffaloes, even in rabbits from this UP, even in Himachal Pradesh and in Tamil Nadu. Then mechanism, how it causing toxicity. These ochratoxins, what they will do, particularly this ochratoxin A, it will cause a lipid peroxidation, and they will generate the reactive oxygen species, and these reactive oxygen species they will cause a lipid peroxidation, and lipid peroxidation is very important. And the another mechanism is they will directly affect to the macromolecules like protein, DNA, RNA, and they will ultimately also form a DNA adduct when they will covalently bind with the DNAs, and they will produce the toxicity. Beside that, it will affect to the mitochondria, and number of studies, even the ultrastructural, we have also seen in number of experimental trials where we given the ochratoxin to the animals, even in birds, that the mitochondria is mainly affected, and it is a non-competitive inhibitor of both succinate cytochrome C reductase and succinate dehydrogenase once they enter into the cells, and it will affect the mitochondrial respiration and oxidative phosphorylation. And that's why, and ultrastructurally, we can see the different mitochondrial forms, and the ring form of the mitochondria, those you can see in this picture, and in this one number of ring form of the mitochondria in the kidneys, you can see. That means the target organ is a kidney, because these proximal convoluted tubules where the OTA receptors, and that's why the PCTs, those all are the mainly affected during the this ochratoxin toxicity. Besides, it will exert apoptosis, and in vitro studies, they suggested that it will cause apoptosis by different mechanisms. Even direct toxic action studied by the people in the different cell lines, in vitro cell lines, and they suggested that the renal cell lines, particularly the PCTs, are the direct cytotoxic action. So this one is a picture from one study where we have given the 3 ppm ochratoxin for 21 days, and in the Japanese quail, and we have seen the retarded growth in the ochratoxin fed birds. You can see the white, these two birds, they are showing retarded, and there you can see the dull, depressed, as well as having ruffled feathers of these birds. However, this one is a SX is the Cibothon. We have given the amelioration, the plant, this lip extract, methanolic extract of the Cibothon, and that is a element for the it decrease the mortality as well as growth parameter the pathology in the birds. Carotenoid pigments in the shank region, particularly that is characteristic in case of fall, gross can identify during the in birds, and this total we have given the rate of for 6 weeks. It can produce the carotenoid pigments, loss of carotenoid pigment. We can find and we can greatly develop toxins. Then see, as I told, it is a nephrotoxic. The kidneys will be swollen and tanning color. That's it is hepatotoxic, so that's why the liver is also enlarged. Maybe emergency you can see for organs.
Sorry to interrupt, sir. Dr. Patil.
Yes, yes.
I guess there's connectivity again. There's connectivity problem. Your PPT is not moving ahead.
Okay. Shall I switch to another system?
Clear. Yes, sir. Please go to the other system. Yes, sir. Switch to the another system, sir.
It will take 1 minute, not 1 minute. Okay, I will see. Hello, Dr. Gauri.
Yes, sir.
This system is okay?
Yes, sir. It's okay.
Okay, again we'll start. No problem.
Sir, can you share your screen again?
Yeah.
Start the slideshow.
So visible now?
Yes, yes, yes.
I think this
Much
one
better.
is much better. That okay. Okay, we'll continue. Sorry for the interruption.
From here, sir.
Huh?
Back. From here, actually, it got struck from that slide.
Okay. So this one is a picture. We have given in the one explanation where we have given ochratoxin A at 3 ppm ochratoxin A for 21 days and in the Japanese quail. And you can see the ochratoxin A, that is ochratoxin A treated birds, OX birds. These are both birds. They are showing the dull, depressed, and the reduced in weight and the ruffled feathers. You can classically, or you can identify even the development of the feathers is also affected of these birds. And the another bird SX group, that means the cibothorn extract, leaf extract, we have given to these birds as an ameliorative agent. And it was showing the potential. It is a partial protection against the ochratoxin induced toxicity in the Japanese quail. Then this another picture where you can see this. This one is also from experimental study. This picture is from experimental study where we have given 2 ppm ochratoxin through diet for 6 weeks in the broiler birds, and you can see the typical loss of carotenoid pigments in the leg of a fowl, and this is a very typical and classical, and you can identify in the birds or in the animals. Then grossly, as I told, this one is a primarily nephrotoxic mycotoxin, and the kidneys are, at the time of postmortem, when you see, the kidneys are mainly involved, and the kidneys will be pale, swollen, or enlarged, and the mahogany to tan in color. And the liver, it is a secondary hepatotoxic, so that's why the liver will also showing the enlargement, pale fiber, and some hemorrhagic spots over the surface.
Even the lymphoid organs and the testes, particularly will cause the atrophy. And as you know, this aflatoxin is also is considered in the endemic regions where it can cause a human testicular cancer. And we have also seen in our studies in the rats, I will show the pictures where the testis is showing the atrophy, maybe unilateral or bilateral atrophy in the young rats when we given exposed given the OTA through diet. Then histopathologically, you will find the degenerative to necrotic changes, and these changes are predominant in the proximal convoluted tubule than the distal convoluted tubule. And these proximal convoluted tubules, number of people they suggested that they are having the receptors for this OTA, and that's why these proximal convoluted tubules they will show the more severely affected than the distal convoluted tubules, besides the thickening glomerular basement membrane, tubular enlargement, even the cast formation, hyaline cast, even epithelial cast, even mononuclear cell infiltration. These all are the changes you can find in the kidney. Besides, liver is also showing degenerative to necrotic changes, even variable inflammatory changes you can find in the hepatocytes and the lymphoid depletion, that is classically you can find in the lymphoid organs. So these pictures from the experimental pictures and 7 days of post-feeding of this aflatoxin in the Japanese quail, and you can see these swollen kidneys. These are the swollen kidneys and distended ureters. We can identify in the whenever the wellness of this kidney, and that you can identify from the control kidney. Then the microscopically you can identify there is a degenerative changes are predominant, and these PCTs those all are mainly involved, and they are showing the increased cytoplasmic granularity, and that indicates, and even the epithelial hyaline bodies also you can identify within the cytoplasm. Then this one is a study in the rats, and you can see the control the kidneys and the aflatoxin treated kidneys, and the color you can find in the necrotic foci also you can identify over the surface of the kidney. Even the testes, as I told, up to 8 weeks, this 4 mg per kg, 4 ppm aflatoxin we are going through diet, and you can see the control testes of the young rats and the aflatoxin treated animal testes, and those are showing the severe atrophy or marked reduction. And even the unilateral atrophy also we have seen in 1 or 2 animals, and that indicates that in humans also why they are correlated in the endemic regions. Besides the nephropathy, it is also affecting to the gonads like the testes in males and may cause testicular cancers. In the 4 ppm level for 6 weeks in the kidneys, you can see the marked degeneration and maculations in the PCTs. Even you can see the chiromegaly also we can visualize within the tubules, and this loss of brush border you can see here, and this one is in the control animals, this brush border you can with the special stain like periodic acid sheet. Then the marked necrosis you can observe in this slide, and the tubules those are showing the necrotic changes, severe necrotic changes or diffuse necrotic changes with the pycnotic nuclei you can find in the most of the tubules. Even the glomeruli those are also affected. However, at 4 weeks of administration, the glomeruli are comparatively normal, and the degenerative to necrotic changes are comparatively less. That means the duration of the exposure of the mycotoxin is also very important besides the dose, and as the duration is the subacute and chronic, the changes will be more intense or the most severe in the different organ system. Even the liver at aflatoxin at the rate of 0.75 mg body weight given, so it can show in the centrilobular pattern of these the hepatocytes showing the fatty change, and that is very classical. And even the malasic foci also in the brain, those are also we are seeing in the gray accumulation of gliosis, that also we are seeing in the brain. And the testes, those testes we have seen. So 4 mg for 2 weeks, you can see the early 2-week exposure of the 4 ppm, you can see the slight changes or mild to moderate changes in the spermatogenesis or the degenerative spermatogonal activity, decrease in the spermatogonal activity in the seen in the seminiferous tubules. But in the 8 week of exposure, the testes showing the severe atrophy of the cessation of the spermatogonial activity, and that is very typical. And even the multinucleated cells also you can identify in few tubules, and some few tubules they are showing the complete cessation of the spermatogonial activity. Then genotoxicity also we have studied, and the single dose of 2.75 mg per kg body weight given to the pregnant rats, and this picture is from my PhD work, where the genotoxic, and I have studied the micronuclear formation, beyond the chromosomal aberrations it will cause, and these are the bone marrow aspiration after the 24 hours of the, exposure to the single dose of the aflatoxin and in the pregnant rats. And beside that, the cystochromatic exchange also it is causing. Beside the teratogenicity, as I told, the aflatoxins, citrinate, even fumonisin B1, these are teratogenic mycotoxins, and what they will produce glass malformations, even the visceral malformations and the skeletal malformations, and those we can see when the pregnant animals experimentally we can produce when we give to diet these mycotoxins to the pregnant animals. They will the pups or the fetus, they will show the various anomalies. So here you can see this one is a aflatoxin treatment given to this fetus, and they are showing the variable anomalies. In this fetus you can see the open eye, then protruded tongue, as well as the omphalocele. Omphalocele we can identify, and these are the changes very classical. And beside the stunted growth or the size, reduced size of the by crown to rump length, you can observe the changes in the fetus when I have given the 2.75 mg per kg body weight aflatoxin to the pregnant animal, pregnant rat on the gestational day 6 and 7. Beside that, it will cause variable, as I told, developing nervous system is another important site or the target site of the aflatoxin. And you can see these exencephaly, mild exencephaly, and these incomplete exposure, these bones, the frontal bones, those are incomplete develop. And you can see variable as per the dose is reduced. You can see the variable, the intensity, that it is dose dependent changes you can find in the aflatoxin. Besides visceral malformations, you can see in this visceral malformation, these are the cervical sections of the hair of the fetuses, and these are the control fetuses, and these are the aflatoxin fetuses. We can see the size, these are showing this normal smaller size, and you can see the dilated because of this dilatation, and this you can see the lateral ventricles, those are greatly dilated, even the third ventricles, those are dilated compared to the normal fetus. Even the microphthalmia you can observe in this fetus by the aflatoxin treated fetus dam. And the kidneys, you can see this one is a normal kidney. These are the adrenal glands, you can see, and this one is a kidney. One kidney is a normal, but another kidney is hypoplastic, and that hypoplastic kidney, that means the aflatoxin is not only having target site for the kidney, and that also we can observe and prove with the by giving this single dose challenge during the pregnancy. Then aflatoxin also induce skeletal malformations, and with the help of alizarin red, yes, staining, we can stain these. It will stain, the alizarin red will stain to the bones and the calcified areas, and those this one is a control showing the different bones which are well developed. And however, this aflatoxin treated fetus, the dams given this on 6 and 7 days of the pregnancy, the fetus showing the abnormal development of these bones, even the abnormal development or the waviness of these ribs you can observe in this treated fetus, even the fusion of these ribs. That means these malformations are because of the single dose that can cause this much effect in the pregnant experimental animals. Then another group of mycotoxins is the trichotoxins, and trichotoxins having a number of members of this group. This is a big family having a T2, hydroxy T2, DAS, DON, DON. These are a number of members, and among that most T2, DAS, and DON, these are the most important members, and they are causing toxicity, and these are mainly produced by the Fusarium species of the fungi. And this toxicity is reported in cattle, pigs, equine, poultry, and it is a potent immunosuppressant, particularly this T2. T2 is a potent immunosuppressive action in case of birds or in turkey, and it will produce the oral lesions, and those lesions are classical. Even in humans, it will cause alimentary toxic alukia. The another mycotoxin is a citridin. Citridin is Aspergillus citrinum, and it is also coexistence, as I told, in the coexistence with the OTA in Balkanic countries or Scandinavian countries where the human population is affected with the nephropathy, and these both are having both having target organ, that is the nephrotoxicity they will produce. And if they most of the mycotoxins in nature, they will not form singly, they will not occur singly, they will be simultaneous, or number of mycotoxins, more than one mycotoxins you will find. And that's why the pathology, you may not get a clear pathology of the toxin in the field, and that's why the natural mycotoxicosis in animals or even in birds, it's very difficult to diagnose on the basis of particular the pathology. Then it is also teratogenic, and it is causing endemic nephropathy. Then fumonisin B1, we have also studied B1 here in our department, and we have conducted a number of trials in Japanese quail, this fumonisin B1, and it is also most toxic than the other members of this family, A1, A2, B2, B3, B4, and it is a primarily hepatotoxic. And also it is a carcinogenic, and it will produce liver and kidney tumors. And the nervous system involvement, that is very peculiar in case of equine or the horses, and you will find the equine leukoencephalomalacia it will cause. And we have also seen the nervous involvement, particularly in our one study, that the fumonisin B1, we have given 300 ppm to the birds, and they are showing the typical nervous signs. And these nervous signs we have correlated with the pathological lesions in the brain, and the malacic foci, and as well as the gliosis we have observed. And this is a typical picture we have observed nervous symptoms, particularly in fumonisin B1 toxicity. Besides it is a primarily hepatotoxic, so that's why the variable foci, multifocal areas of the necrosis you can identify with the variable infiltration of the heterophils, even the mononuclear cells after the 40 days, even the 21 days, even the more this cognitive type of necrosis you can find in this liver. Then we'll switch to the diagnosis, and diagnosis, as I told, diagnosis is the most challenging job in the mycotoxicosis in animals, and particularly experimental diagnosis is easy, but the natural mycotoxicosis is very difficult in animals, because as I told, the mycotoxins, they are not these fungi are ubiquitous, and one single fungi can produce number of mycotoxins, or the single mycotoxin produced by the number of fungi. And that's why in nature you may get multiple mycotoxins simultaneously, and they may have, because you might be knowing the mycotoxins having their interactions. If the more than one mycotoxin is present in the food and feed, they may interact. Once they enter into the body, they may interact, maybe they interact in a synergistic way, or antagonistic way, or less than additive effect. That's why the more than additive, less than additive, synergism, or the antagonistic way. And that's why on the basis of their interaction, the pathological changes you can find in. So diagnosis is generally based on the history, clinical findings, or clinical manifestations. Besides, you can correlate, we can take the help of clinical findings, the serum samples, if the liver function test, kidney function test, and those all are indication of the whether nephropathy is going on primarily, or hepatitis is going on, like in aflatoxicosis or nephropathy in octotoxicosis. So we can correlate with these serum findings as well, and those all are also we can use for the diagnostic criteria. Even the pathological finding, as I told, gross, microscopic, even the ultrastructural pathology, those all are peculiar for we have studied in experimentally, and those all are also giving an indication of this mycotoxicosis in animals. But the most the definitive diagnosis is always achieved with the help of the analysis of the mycotoxins in feed and water samples. And nowadays the main research, because you might be knowing the pressure of the change in the climate, climate and the population, increasing in the population, and that's why the people are more concerned about to know how much contamination in the feed and fodder which we are giving to the using for the human or animal population. So these analysis of the mycotoxins includes standard techniques, rapid screening techniques, and emerging techniques. The standard techniques mainly the chromatographic techniques, and it involves TLC, HPLC, gas chromatography. Then rapid screening techniques, those are very useful, and on-site we can use these techniques, these rapid screening techniques, and these are mainly immunoassay based techniques or the biosensor or biosensor based techniques. Those are very useful for the rapid screening techniques, and nowadays for the import-export purpose, the people are using on-site these rapid screening techniques. And another techniques, those are emerging techniques or researchable techniques. Those are newer techniques, and although they are having good accuracy, or they are simply or easy to identify the mycotoxins in food, but their validation is needed. So we are going to talk about those important points about these techniques. So standard or reference techniques, as I told, chromatographic techniques includes thin layer chromatography, gas chromatography, liquid chromatography. Although thin layer chromatography is the oldest method, and the thin chromatography is till date we are using, and it's a very important technique for the quantitative confirmation or semi-quantitative or quantification of these techniques. And these are different protocols and different mycotoxins where these we can detect with these techniques, and this technique till we are using for the detection of these mycotoxins. Then thin layer chromatography is having a rapid analysis, several samples we can do in a short period of time. It's a low cost technique and easy to perform. However, disadvantage is having low sensitivity and reproducibility, and need of large quantity of solvents, and difficulties in automation. So that's why this TLC is although it is we are using, but not practicable, and in the some instances. Then the gas chromatography, the main advantage, it's high specific and sensitive for all the mycotoxins, and we can do the simultaneous analysis of number of mycotoxins by this gas chromatography. But the disadvantage of this gas chromatography, it is not suitable for non-volatile or polar mycotoxins, and that's why it is not suitable for these most of the mycotoxins. Only trichothecins, those are well suitable for these mycotoxins, and the deoxynivalenol, fumicin, trichothecin, those are mainly mycotoxins. Those are we can go for the gas chromatography. Then liquid chromatography, high performance liquid chromatography, and this method is very important and having good sensitivity and versatility. But the most important disadvantage is its portability, and on-site screening, whenever particularly import export or the trading purpose, these methods are not suitable, and the cost, even it need skilled personnel to carry out the analysis, even the methods. Those are very important, and again the time consuming. This one is method. Then HPLC coupled with the fluorescent detection, UV visible and photoiodide array. So these are used even the liquid chromatography coupled with the mass spectrometry, even the tandem mass spectrometry. Those are the nowadays people are using for a number of mycotoxins, for the screening of the number of mycotoxins, and these HPLC we can use for number of mycotoxins, wide range of mycotoxins, to detect the mycotoxin levels in different food and feed commodities, even the juice or the wine and coffee, the other commodities as well. Then rapid screening techniques, among the rapid screening techniques, immunoassay based methods, those are nowadays getting more popularity, and they are because of they are very simple, they are easily cheap, and they are sensitive. And sometimes disadvantage like that, the matrix effect or the non-specific interactions, that's why they are not suitable. But otherwise, it's the most important techniques, immunoassay-based techniques, and a lot of commercial ELISA available in the markets, and we are using those ELISA for number of mycotoxins for the quantification of these mycotoxins, and they require this advantage of the ELISA is a minimum cleanup procedures like chromatographic methods. They require more cleanup procedures and the sample preparation. And the disadvantages, they lack the precision when the mycotoxin is at low concentration, because it is suggested that all the food commodities of the world, those are contaminated with the mycotoxins, but how much concentration is the most important, and that's why the permissible limits are given by the authorities like USFDA or FAO. They have given the permissible limits and for the different mycotoxins, and beyond the permissible limit, these food and feed commodities are not suitable for the human or animal consumption. So that's why besides commercial kits, as I told, and they may have another problem is the false positive, false negative results, and that's the problem. Then we can, these are the protocols by which the different mycotoxins we can detect in different commodities by ELISA. Then red immunoassay is another very important, and it is the first immunoassay technique developed, and nowadays we are also using for the detection of the mycotoxins in feed and fodder samples. The advantages of this method is the multiple analysis we can do simultaneously, and they are having high levels of sensitivity and specificity. Where the disadvantage, because we are using the radioisotopes, and those are the maybe potential hazard for the personnel. Those are using this and even the storage and disposal of those after using those radioisotopes, and that is a problem. Then another one important this rapid diagnostic method is a lateral flow immunostrips or immunodip strips. And these immunostrips or the immunodipsticks, those are used on the basis of pregnancy test kit, like pregnancy test kit, where we can use the load the sample, and it will give the color formation and the test line and the control line if the mycotoxin is present, so it will give the line. And it is very useful and very easy, and it is a portable, and it is a rapid test, and within few minutes we can get the result, like pregnancy test kit, whether the animal or the individual is pregnant or not, we can use. So similar way, this one is also, and this one is also disadvantage is reproducibility and sensitivity problem with this lateral flow immunoassay. Then fluorescent polarization immunoassay, this one is also important, where you can use the rate of rotation of a fluorophore, that is a tracer we are adding beside the antigen antibody, and the more tracer we can find whenever there is a no toxins, and the less tracer you can find bound you can find when there is a toxin is present in the test. And the fluorescence polarization single value inversely proportional to the amount of mycotoxin present in the sample, and we can identify. And this method is a reliable, rapid, easy to perform, and disadvantage it is a more expensive, and a lot of research is going on for the to cut the cost of this technique. Then another one is important after these immunoassay techniques, the biosensors or biosensor based methods. Those are also very useful nowadays, and the number of biosensors, even the nanoparticles, also nano-based materials, those are also coupled with these biosensors. They are also helpful for the detection of mycotoxins. Not only mycotoxins, even the these multiple mycotoxins, even the fungi, pathogenic fungi also we can detect. And these are having the piezoelectric quartz crystal microbalances. So that one is a piezoelectric, optical, or electrochemical sensor. Those all are useful, and those all are available in the market. And these all are having advantage. These techniques are having a fast result, quick result you can get. They are having low cost and having greater sensitivity, and main advantages, they are portable. However, disadvantages, they require specialist skilled personnel in analytical equipment, and low selectivity and reproducibility. Those are the other problems. Then emerging technologies, nowadays number of techniques, those all are coming, and a number of research is going on to detect the mycotoxins at a low level in the food and feed communities, and the people are trying to develop new and new technologies. And among that, the molecular techniques like PCR-based techniques, the proteomics, genomics, then the electronic nose, then quantitative NMR techniques, then hyperspectral imaging, and aggregation-induced emission dye. These are the techniques we will see one by one. So proteomic and genomic methods, those all are useful, and these are PCR-based, maybe PCR, quantitative PCR, LAMP, RT-PCR-based, and even proteomic methods, mass spectrophotometric technique. And although these are the expensive, comparatively expensive, but the main advantage of these techniques are you can detect the fungus as well as the mycotoxin, the pathogenic fungus. Those are producing the mycotoxins. We can detect with the help of this genomic and proteomic analysis, and we can detect these techniques, detect these mycotoxins and the toxicity fungi. Then electronic nose is based on this gas chromatographic system, where these volatile compounds, these mycotoxins, these volatile compounds, and these they are giving the characteristic odor, and those are with the device, proper device, we can use. And this is a like mammalian olfactory, in our nose, olfactory receptors, then also olfactory bulbs, and then signal pass to brain. So like that, we are also sensory arrays, then signal transducers, and then pattern recognition engine we can identify, and with the help of device we can identify the fungal metabolites in the feed and fodder. Then aggregation induced emission dye technique. This one is also fluorescent based method, and you can see the aggregation induced emissions. It will cause the emission and the normal solution, and this aggregation based they will produce intense fluorescence, and that intense fluorescence, and with the help of that fluorescence we can identify, or we can detect the mycotoxins. And octotoxin, the actor sensor, using this AI dye-based actor sensor, it is useful for the detection of octotoxin that is developed. Then the quantitative nuclear magnetic resonance, that is also a basic technique, and it's an analytical tool for quantitative analysis of mycotoxins like fusarium. And advantages, these techniques we can use without these standards, and the reliable identification of isolate metabolites. And disadvantages are they are low sensitive, and having low commercial software or limited quantification methods. Then another last technique, there's a hyperspectral imaging. And hyperspectral imaging, that is a spectral reflectance of each pixel is acquired for range of wavelengths in the electromagnetic spectra. And you can see in this picture, these electromagnetic spectra, I mean, they have different wavelengths, and there's an imaging component, and it is a rapid and non-destructive method for the analysis of the fungal and mycotoxin assessment, and people are using. Although the disadvantage like cost effectiveness and the multivariate algorithms, those all are required for this method. Then we have seen the diagnostic aspect. Then we are switching to the management, and management is a key because the management and the management of mycotoxicosis, because these fungi are ubiquitous, and they will contaminate, and the number of practices, particularly pre-harvest, during harvest and post-harvest methods by which these fungi they will contaminate the crop and ultimately agriculture commodities, and through the cereals or the grains, and they will enter into the food channel, into the animals and humans. Then the pre-harvest management includes control of insect infestation, crop residue, crop rotation practices, irrigation, soils conditions, and development of resistant plant variety. These are the methods by which we can the pathogenic fungi we can avoid, so that the like insects, if you control the insects, they will not damage the grains, and ultimately the fungus will not allow the fungus will not get an opportunity to contaminate the grains. So that like the pre-harvest methods we have to go. Then harvest technologies include the agriculture products with timeliness and cleanup and the drying of these crop. Then the quick harvest, that whenever the cycle is completed, the fully grown crop is there, then we have to go for the harvest into more humidity, to more moisture, and the moisture it will cause the favor the growth of the fungi. And the temperature, is below 5 to 8 degrees Celsius, so we have to keep, so that the most of the fungus, they will not grow this temperature. And the moisture more than 11 or 12%, if moisture is there in the grains during the storage procedure, so that time also the fungi will get an opportunity. So that's why we should keep below the 10-degree moisture. Then the post-harvest technologies include the decontamination strategy, detoxification, or amelioration, because the mycotoxin contamination is there in the feed or food, and we have to destroy that mycotoxins, and we have to see the criteria for those, that is inactivate, destroy, or remove the mycotoxins, no residues of toxin substances in the feed or food, and it retain nutrient value, because the nutrient value should be maintained. Whatever procedures or techniques we are using for detoxification methods, we are using for the post-harvest or amelioration methods, that should not reduce the nutritive quality of the feed and fodder, and that should be acceptable. No alterations of the products, technologies, or properties, and they again further destroy the fungal spores. Then post-harvest management includes mechanical or density separation, drying, irradiation, processing of food, antimycotic, use of antimycotic agent. Number of antimycotic agents are there, so that also we can use when processing of food, like with milling, roasting, autoclaving, malting, brewing, cooking. Even simple cooking also reduce, even the heating, that is also will reduce. Even water treatment, that will also reduce the mycotoxin levels in the contaminated grains or in the food. Then mechanical and density separation, like in groundnut, whatever the affected groundnuts, we have to separate it. That is usual practice. And when the drying, ultraviolet, the irradiation, when the drying, simple sun drying is also, it will reduce the mycotoxin level in the feed. Then chemical methods like structural degradations by using acids, bases, aldehydes, then other substances, even gas. Then ammunition. Ammunition is the most important technique or chemical methods by which using the anhydrous ammonia gas, liquid ammonia, by which we the mycotoxins, the level of the mycotoxins like aflatoxin and ochratoxin we can reduce. Even ozonization, ozones are more the oxidants, the potent oxidants, and the sodium hydroxide treatment is also, it will reduce the amount of mycotoxin level in the food and contaminated food and feed. So that's why physical methods, those are having, although they are reducing the mycotoxin level in the contaminated food and feed, but they are not maintaining the quality or the nutrient quality of the food is not maintained during this chemical processing or the physical ways. So that's why amelioration is a strategy, and nowadays people are trying to go for the ameliorative agents. Number of ameliorative agents, those are mainly categorized as a mycotoxin binding agents, and even commercial mycotoxin binders. Those are available, and we are even the commercial feed prepared by the standard companies. They are adding this mycotoxin binder in their feed. Those are used particularly for maybe used for in the poultry or in the livestock feed. Even microbiological binding agents, even antioxidant substances, even feed components and additives. Those are the other ways by which we can do the amelioration. We'll see the one by one those amelioration strategy. Those are available and people are using. So the use of mycotoxin binding agents, among that these binding agents, what they will do, they are nutritionally inert, and what they will do, they will reduce the bioavailability, because this amelioration is basically immune strategy. However, the physical and chemical methods of the detoxification, those are in vitro activity. And this HSCS, hydrated sodium calcium aluminum silicate, those are used and showing efficiency against the aflatoxicosis, even the other mycotoxins, and they need to be trichothecins, and they attract the polar structures of the various mycotoxins, and in broilers particularly 0.5% HSCS, it can cause less effective against the OTA than the aflatoxin B1. Then bentonite or the clay, it is an absorbent aluminum phyllosilicate clay, and this bentonite, particularly sodium bentolite, once it enters into the body, it will be swelling, it will swell, and it is more effective than the calcium bentonite, and it will having the layered crystalline changeable cations, and they are found to be an effective against aflatoxin, and that's why European Commission in 2013 they have allow it is and they found that it is an effective against the binding mycotoxins, especially the aflatoxin and bentonite. And then nowadays people are focusing on the work on the different forms of the bentonite, like dioctahedral, bentonite, and those all are being used for the commercially against aflatoxicosis. Even they are having good potential again in vitro studies against Genotoxicity and Fluorescent B1 as well. Then clays like phyllosilicate minerals, and they are also being used as a mycotoxin binders. And these lacks toxicity, contains high surface area and cation exchange capacity, like zeolite, kaolinite, montrimodionites, then the vermiculites, mentorites, and monticollites. They are having variable properties, and they are more useful against the aflatoxicosis than the other Zen or OTA. And this nowadays in recent report is in Holstein cow, this were vermiculite, mentorite, and montmorillonite that reduces aflatoxins level in the rumen, milk, and feces, particularly in the Holstein cow. Then another one is activated carbon or charcoal strategy, and although this now recent report, it is suggested in vitro having a good binding ability against several mycotoxins, but in vivo recent report of Patil, they suggested that it's a limited absorptive potential against the aflatoxin A. Then cholestyramine also ion exchange resin, and it's having strong affinity for the bile salts, and it protects the rats against OTA-induced mycotoxicity. Then 5% use of the cholestyramine in diet. It can reduce up to 50% of the OTA concentration, plasma concentration of OTA. Then in ruminants, it is an effective absorbent of OTA in the GI tract, and it's having potential for the streptomycin and Genotoxicity. Then polypyrrolidone, this one is a synthetic resin, also showing the partial amelioration effect. Then the bovine serum albumin, it's having in vitro binding ability with the mycotoxins, and it competitive inhibition. That's the way it is used as a mycotoxin binder. Then microbiological binding agents, among that manino-oligosaccharides, because these Saccharomyces cerevisiae cell wall, this yeast, they are having very broad spectrum efficacy against most of the mycotoxins, and having more efficacy against the aflatoxicosis compared to aflatoxicosis. And besides, what they will do, what it will cause, it will give this stimulate the immune system, and that's why it will increase the IgA and IgG levels when given the 13.11% dosage. Then beta D glucons of the yeast cell wall. Now this recent report of Azami, they suggested that 3 grams per day of the beta D glucons from this yeast cell wall. It is an excellent binding potential for the aflatoxin B1, and it can bind more than 90% in dairy goats. Then probiotics and prebiotics. Prebiotics are also using the bacteria. These Bacillus subtilis, even the lactic acid bacteria, bifidobacteria, Mentococcus groups of the bacteria. Those are having affinity or the binding ability with the mycotoxins, and they limit the mycotoxin bioavailability, and that's why they are reducing the toxic effects. Then antioxidant substance, basically these antioxidant substance, they are, as you know, particularly the vitamins, vitamin C, vitamin A, vitamin E. These are the very useful antioxidants, and their main action is they will free radical scavengers, and most of the mycotoxins like aflatoxin A As I told, they will release powerful reactors, oxygen species when they enter into the body, and that's why they will produce this damage to the plasma membrane or cell membrane, and that's why the free radical damage they will produce. And so that's why the ascorbic acid is having a quenching effect of the superoxide hydroxyl radical singlet oxygen, and they will partially ameliorate the OTA toxicity in layers, even in the rats. Then vitamin E and vitamins, the selenium with or without selenium. So these are also useful, and they will alleviate the toxic effects of aflatoxin B1, and they will reduce the oxidative stress during the mycotoxic exposure. Even vitamin A is also anti-mutagenic, anti-carcinogenic because the presence of the carotenes and xanthophylls. Then food components and additives, and they includes the like phenol. These are structural analog phenol moiety of the OTA, and the aflatoxin competitively inhibits phenamine tRNA synthesis and phenamine hydroxylase during the toxicity. And that's why if you give L-phenamine therapy in the diet during the aflatoxicosis, so it will reduce the mortality rate, and we have also the reports also suggested that the poor people toxicity, and the addition of 0.8 to 2.5% of the L-phenamine addition in the diet, that will reduce the mortality rate. Then methionine, aspartame, these are also being used and ameliorating agents. And these aspartame inhibits the OTA-induced cytotoxic effects and ultimately protect the animals. Then food protein, the protein level, if you increase during the mycotoxicosis, that in the diet, or maybe from 14 to 18% to 22 to 26%, that will also alleviate the toxic effects of the aflatoxin-induced toxicity in birds. Then dietary lipids, the lipids interfere with the absorption of the mycotoxins. So if you increase the dietary lipid levels, that will also alleviate the toxicity of the mycotoxins. So we have seen the various mycotoxins, their diagnosis, and their amelioration strategies. And on the basis, I can conclude that the mycotoxins, these fungi, toxigenic fungi, those are producing the mycotoxins, they are ubiquitous in nature, and the mycotoxins having diverse toxicological spectrum, and that's why they are posing potential threats to the animals and human health, because their carryover effect or to enter through into the food channel in the form of the residual effect to the food and the animal byproducts. Then the diagnosis of the natural mycotoxicosis is often difficult. Experimental mycotoxicosis we can diagnose, but the natural mycotoxicosis, where the number of mycotoxins they are producing simultaneous, simultaneously, they are responsible for production of the toxic effects in the animals. So it's very difficult because of the pathology or the other clinical manifestations. Those are not supporting to the detection level. Then the level and duration of the exposure of the mycotoxins and the health or immune status of the animals, those are also most important, and that's why diagnosis is always difficult. Then the definitive diagnosis, for definitive diagnosis, we have to go for the detection and quantification of the specific mycotoxins. Those are in the contaminated feed and water samples, and for that we have to use precise sampling technique and the appropriate mycotoxin analytical methods. And nowadays emerging methods, number of methods are there, but we need to validate those methods, although number of options or number of techniques are day by day novel techniques, those are available in the market. Then physical and chemical methods, as I told, those are impracticable in terms of safeguarding the nutritive quality of the feed and food, while the ameliorative strategies which are showing promising results in the management of the latest effects of these mycotoxins and mycotoxicosis. Okay, thank you.
Thank you, sir. So now we will move to question answer session. We are having one question that is to control non-adsorbable adsorbable mycotoxin in poultry feed. Which control measure or product is working well?
What question? Can you repeat, please?
Sir, to control non-adsorbable mycotoxins in poultry feed, which control measure or product is working well?
Which product? Because the mycotoxins, not a single product is useful, because as I told, number of the strategies, dietary strategies or the emulsion strategies, those all are used, but those all are having variable results in different species as well as in the with respect to the mycotoxins. So I can't say which product specifically.
Thank you, sir. Is there any other question? Dear participants?
Okay, I think there is no question now.
Yes, sir. There is no question. Thank you, sir.
Okay, okay, Dr. Gauri, thank you so much. I'm thankful to the ICR IVRI as well as NABCL Baramulla for giving me this opportunity. I'm especially thankful to the Dr. Suresh Radotra for giving me this opportunity. Thank you so much, sir.
Thank you, sir. Thank you so much. You may leave now, sir.
Okay, okay, thank you.
Okay, thank you, sir. So in the afternoon, we will be having Dr. Pawan Kumar, Division of Pathology from IVRI Izzatnagar. So meet you at 2:30. Thank you so much.